Right, okay. So my name is Annie and I work at SSERC, and right from the beginning, I probably have to make sure that you're happy with what you're here for. So we have had a huge interest in tonight, and you know, I think we know why that is. The return of the assignments is causing a lot of anxiety, and with SQA and their Understand Standard events being a wee bit later, what I'm here to do is really about practical work, and I'm going to try and stay clear as much as I can from the SQA side, but there's wee bits and pieces that I'm going to add in, okay? So I've disabled mics and camera because I'm worried about it lagging, but otherwise the chat does seem to be working. So you please feel free to add things in as we go through. What I'm going to try and do as best I can is cover National 5 assignments with bits of higher. I've marked for a number of years National 5 assignments, but I have never marked higher biology or human biology, so I'm not going to touch on that. But I've got things in place if people did want to share hints and tips about either of those, because I recognise that a lot of people will be here hoping to hear more maybe about that side of higher. So the programme that Jane would have sent out to you through the week had these different sessions that I'm hoping to run through. So different practical activities that across all of them, they will work for Nat 5, but in places for higher and higher human, and I'll highlight those as we go through. I've put time for questions. I think realistically with so many people in, that's going to be really difficult. So I've got a Google Doc that I've got there so that you can add questions in, and if I don't get to them, I will absolutely try my best to answer them as best I can, or to pass them on to SQA, who said that they will answer them. So what to expect? So I'm going to give a description of the practical work that I'm going to suggest, which is based on what SQA have suggested. In this particular presentation, I've sort of made condensed videos because I thought it's going to be really tiresome for you to listen to me going, and then step 1, and then step 2. So I've condensed them, but the full PowerPoint and the lab book that I'll share with you at the end has all the steps in them. So don't worry if it feels like it's way too brief. I have indicated at times some marking support. So particularly for only for National 5, because I don't know anything enough. I've never marked higher, so I can't give any advice on that. So as I go through each individual practical, I'm going to take one part of the marking instructions and say what I've learned over the years of doing that, or what a good answer might look like. And please do feel free to step in and say, Annie, that sounds absolutely rubbish. However, I'm supposed to say at this point that that would be absolutely SQA would be the people to go to for that. So I don't want to step on toes there. There's a big number of people on the call, so just in case, I'm going to show a couple of QR codes that I've got for Google Docs that are open for editing. One of them is to add questions in if you feel that the chat isn't working, or I would actually almost prefer them in there, because I can review them all together, and SQA have asked that I pass on any questions that you had, and I can filter them and things, but they want to draw up an FAQ, FAQ based on that for their Understand Standards events. And yeah, if there are issues, please do feel free to not stay frustrated and just leave. And if I notice that people have dipped away, then I will sort something else as an alternative. Okay, so here is the Google Doc for the for any questions or comments that you have. So please feel free just to have that open. And can there be a link as well? That interest now. And then, oops, I have another one as well. So let me just try and do that quickly, if I can find it. So the link for this one, if I find the chat, I can post. So this first one is for the questions, and the second one I'm going to put in is just hints and tips. So if you as we go through the marking instructions, when you go to these marking events, sometimes you get little wee side notes. Oh, that's what this sort of means. And I've over the years taken little notes, and there's not huge amounts, but it might be that you find it particularly useful. So they are there. I'm still sharing my screen, so I'm hoping I can just go back in, okay? Okay. I don't like that so many people have left. I hope this is okay. Okay. Right, let's get started. So the first thing that I think is, if you're maybe new to if you're new to doing the assignments this year, or it's been such a long time for everybody, if you've forgotten or don't know that this page exists, these are absolutely essential PL for you in your own time. So SQA have got 4 on National 5, they've got 4 or 5 on higher. You can download the script that a candidate has submitted, and then the commentary bit is where what mark did they get? Did they get the mark or not? And if not, why not? So it really helps you understand exactly what you have to do to score well. And there's good ones in there for what we're talking about tonight to sort of add to what we're talking about. I contacted SQA before we started, and their key message to everybody is that the coursework documents have not changed since the last time it was delivered. So in 2019, 2020, they are the same rules apply. So that's their party line for every single subject. Yesterday as well, they released a range of different dates across Aberdeen, Dundee, Edinburgh and Glasgow for different Understand Standard events that are in person, and then they're going to release the online webinars soon as well. So when you get this PowerPoint, you can click on all those links there and book your places on those. There was another couple of things in those documents for the mark instructions or the assessment task document that I thought, I feel it's a bit ambiguous what that means. So this was the first bit. Choose with support a relevant topic in biology, and there's other bits underneath that. And I thought, what do you mean by a topic? If you've got a class, can they all do enzymes? Or what do they mean by that? And so SQA said, yes, they can all do enzymes. That would be classed as a general topic, but they have to do different independent variables, which is exactly the same as how it was. So that's cool. So they could do enzymes, look at pH, temperature, substrate concentration. Okay, I will come back to that. But they can also look at different experiments or ways of doing that experiment. So let me just see if I can get these people in. I'm not seeing that at all. Okay, can't see it. Okay, so for them to meet the conditions of the assessment, they either have to work individually or they can work in small groups of 4, depending on what materials you have. But they have to apparently decide on the range and the interval of the values of the independent variables. So if you had substrate concentration, they're saying the candidates should be choosing that range and interval and investigating different aspects of the topic, which I would be saying is an independent variable, and or use different methods. So there's various ways as we go through this that will highlight the different methods that can allow you to work within that guidance as well. These are the documents that I've basically been working with. So SQA release a range of different, their recommendations for possible investigations that you can do. And over tonight from the National 5 one, I'm going to look at pectinase. As soon as I put highlighting on, it never is happy moving on from this. Catalase, and I'm going to look at concentration of substrate, transpiration, and then using algal balls to look at photosynthesis. So I'm going to cover those, but there are a whole range of different things that they're happy for you to do as well. The marking for the National 5 is up to 20 marks, and this is all the different aspects of the assignment that are going to be marked and the points, the marks that are awarded at each of those different sections. So we're going to cover most of those, not all of them, as we go through. So the first experiment that I'm going to look at is the effect of an independent variable on catalase activity, and for those independent variables, they have suggested the usual suspects at National 5, okay? And this would also work at higher if you were looking at different options there. And, oh, that was higher human, and this one for higher. So they're basically exactly the same. There's different methodologies we can use for this, which is really nice. So even across a room, they could, instead of changing the independent variable necessarily, they could actually choose one of the methods if that was easier to set up. So they can look at height of foam, which a lot of people have done. This would be the typical way that I would have done this all the time. They can do displacement of the gas, where you've got your beaker of, or your conical flask of yeast and sugar, or your hydrogen peroxide, and you're going to collect the oxygen gas in an upturned measuring cylinder of water. Or you can do mobilization, and you can either immobilize the tissue, and you're going to look at how long it takes for the yeast bead, in this case, to fall and rise, or you can just take a filter paper disc, because time is really tricky. If time is really tricky for that to work, then you can just soak the filter paper disc in that liquid as well and drop that in. So a quick background if it's been a while since you've done this. So catalase is an enzyme that is going to break down hydrogen peroxide, and we're going to vary the concentration of that, and it's going to release oxygen and water. And in this particular version, we're going to mobilize the yeast with sodium alginate, and once the beads are prepared, we're going to drop them into a container of hydrogen peroxide. So I'm just going to show you this, this video here. If someone can let me know if you can't hear the actual volume, and I'm going to just check all the stuff in the chat whilst this is running.
In this video, we're going to look at the mobilisation of yeast to measure catalase activity. These are the range of materials that we need for the immobilisation stage, so working with 10% yeast suspension and 2% sodium alginate. For the first part, we're going to look at this as an overview, and we've got our mixed yeast and sodium alginate suspension, and we're going to drop that through into a calcium chloride solution. So you'll start by mixing those two materials together, and then pouring that into the barrel of a 10 cubic centimetre syringe, and allow that just by gravity to drop in bead by bead into the calcium chloride. And that will continue until all of that mixture has passed into the calcium chloride, and you stir that or agitate it periodically. And once those beads have allowed to settle, we're then going to use them in stage 2, which looks like this. So we've got different concentrations of our substrate from 0.5 volt to 2 volt. And in this, we're going to rinse off our yeast beads with distilled water. We drop it into the universal containing the hydrogen peroxide, and we're going to time how long it takes for that bead to fall and rise, and repeat that with the various substrate concentrations. And as you can see here, this has been repeated in triplicate with mean values displayed, and we can see a negative correlation between increasing concentration and the time it takes for that reaction.
Okay. Right, there are some technical issues, and I'm really sorry for some people who are having some challenges with it. I'm not going to go back into those docs and chain. I'm not sure what's happening with those. So if you're struggling to ask questions, please just add them in there, and I'll have a look at it afterwards. Perfect, great. So when we've done this experiment with that particular concentration of yeast that was in that first slide, and you'll get it in the lab book at the end, these are the values that you get with yeast. And in my experience, one of the first times we did this in school, we used, based probably on our second source, we used things like apple and carrot, and it was taken like 5, 6, 10 minutes for these beads to come back up to the top. Yeast is brilliant because it's really fast. And in that bottom one with 2 vol, really low concentrations, it also keeps it nice and safe. You're not working with high concentrations here, the substrate. Then it's working in like 10 seconds and super reproducible, as long as you've got a bit of an issue with the human effect, which might be a good point for evaluation with the drop time and the reaction of that but it is very reproducible. So it does keep that sort of that variability within 3 repeats is fine. Okay, let me see, we can change time. No. So with the reaction time thing, I'll come back to your questions if I'm struggling to keep up with them all, but and for the yeast speed, yes, I use a new yeast speed every time, but technically the enzyme should never run out, but I always do. I always just, because actually here, if you think about it, for each group, all you're going to need is 9 yeast speeds. And once you've immobilized, you get a good number, but they could just be rinsed and passed to another class. It's not going to, the enzyme never gets used up. So that's also a really good benefit of that. The other way of doing this is not to immobilize, although it's a really lovely technique to do, and just to get a filter paper disc that you've hole punched, and then you soak it in the yeast, and you do exactly the same. And it's also really lovely, and just get a pair of forceps, whip out the disc, and then repeat it with a new one. So those are the kind of values we're going to say. In terms of reaction rate, that is the issue. And yes, you use the same container of hydrogen peroxide every time. That's also the lovely thing. With the height of foam, you have to pour it out. You've got all the cleaning to do, and it's messy, and it uses tons and tons of hydrogen peroxide. And you also get all the horrible unreacted hydrogen peroxide on skin, which, if you've got skin like me, ends up burning and it's sore. So this is lovely. You just take the bead out, put a new one in, same hydrogen peroxide each time. And the reaction rate thing is just an issue with this that you could talk about, I guess, in the evaluation. I'm not entirely sure, thinking off the cuff, what you would say, but that's definitely the limiting factor here, that you're relying on people having the same reaction time when they do that. Okay, and yeah, that's just to show what the graph looks like if you do it on Excel, which I will come back to later. If you're moving further up the school and you wanted to introduce inhibitors, we've got this bulletin article, which is in the folder that I am sharing with you. And there, we'd previously done it where we took the yeast and the copper sulfate and immobilized them together, but that doesn't work. Alginate does not like salt. So you take your beads and you incubate them for like overnight in copper sulfate, and they become a little bit blue. And then you can use them, but don't over rinse, because then it comes back out again. But this bulletin article talks about how you can look at inhibition using this process as well. Okay, if you use the same bead and don't change it each time, it gives an about. No, if you use the same bead, that's absolutely fine, because your enzyme should be okay. I would probably take it out, rinse it, and reuse it, but I usually just get a fresh one every time. But I don't think it would be an evaluation point, to be honest. I mean, I'm not an expert. I guess maybe you could ask SQA something like that. So at the end of that, I'm going to start with each one just taking a few bits of the marking scheme, and this is a nice easy one to start with. So a name that describes clearly the purpose of the investigation. We don't need to use the word aim, but it should tell them basically what is going on. And these bits here actually would be a really good title, but not enough for an aim. So something here like, to find out how concentration of substrate affects rate of enzyme activity would be good. So you're not mentioning hydrogen peroxide or catalase, which would also be fine, but this more generally is also fine. So there you would get a mark for your aim here. When you're talking about underlying biology, this is marked more, the key thing here is that first sentence, expanded descriptions and explanations. So they don't want, which is down at the bottom, a simple statement. So you're not going to get a mark for enzymes or biological catalysts. So as an example, I've pulled this from an old Understanding Standards document. And each of those, I used to always say, make them into distinct paragraphs where you think you've explained something enough. Have you described what an enzyme is? So that first opening paragraph talks about what an enzyme is, talks about the shape, the amino acids. That's all cool. That's all relevant to what they're talking about. So that's expanded description about what is an enzyme. The second bit goes on almost talk about specificity, the active site, and that's also expanded. So those individual sentences on their own isn't enough, but at the end of that whole paragraph, SQA said, At that point, I would give them a mark. And the same for the last bit. Their aim is about substrate concentration. This last bit, they've not talked about temperature, they've not talked about pH, they've focused on substrate concentration because that's what their aim is. And so at the end of that paragraph, SQA said that would be a mark. So that's sort of the level of detail they want. If they talked about temperature there, in my opinion, my experience, they wouldn't get a mark because that isn't relevant to what they have been talking about. Okay, so I can't remember how far into 3 I go, but this is now looking at a brief description of what the protocol was. And all SQA are looking for here is, can they visualize what this actually looks like? I don't need to be able to repeat it, but I need to be able to see it in my head what this looks like. And they've got great examples at the bottom to sort of give you a feel for the level of detail, and it's totally counter to what you're used to doing, but that's all they have to do. So an example that I said for this, which maybe arguably is a little bit too much, but because we've done the immobilization, I've got a really brief statement about what we did. And then we took a bead, we dropped it in hydrogen peroxide, and we timed how long it took for it to sink and rise. Okay, not rise and fall, sink or fall and rise, has to be the right way around. That would be a mark. This wouldn't be a mark. So basically, as soon as you look at something and there's numbers all over the place and percentages and times, don't let them do that. So that's too much. This and every other thing that we've ever done, this is great, but not for showing the ability to summarize an experiment. Okay. Yes, basically no measurements, volumes, concentrations, exactly. And if you were going to do something about enzymes, candidate for evidence on that Understand Standards website I showed at the beginning, yes, they will lose the point if they give too much detail, absolutely. But this candidate evidence will give you a really nice feel for what an assignment about enzymes might look like. And so you download the candidate response and you download the commentary, and it's really helpful as you work through those. Okay, next one is transpiration. So again, there's a range of different independent variables that you could choose for this, and it's on SQA's list of what you might want to investigate, and these are the different independent variables they've suggested there. So there's the obvious curriculum content that you can have them elaborate upon in their underlying biology section. So what is transpiration? What processes are actually involved? And then how do those different factors affect the rate of transpiration? And we've then got to think about how do we adapt the experiment to allow for these factors to be investigated. So I'm going to show you this one. Now, as a wee warning, I find, I'm sure lots of you do, transpiration really difficult. And you'll see as you go through, and in the things that I put into the folder at the end, it's fiddly, there's no getting away from that, but it's also cheap as chips to set this up. So if you've got, as seems to be the situation, hardly any cash to do anything, you just need to make sure you've got a good tree nearby that you don't mind taking leaves off every now and again, and some tubing, a clamp stand, and a hairdryer or something, and you're pretty much good to go. So you could give this a try, and SQA have suggested this as something they would recommend, obviously just for National 5.
So in this experiment, we're going to look at investigating transpiration rate in horse chestnut tree leaves. And this is a suggested possible assignment topic in National 5 Biology from SQA, where you can look at wind speed, temperature, number of leaves, or the surface area of the leaf. This fits into the curriculum where we look at what transpiration is and how water moves through a plant, and the various external factors, including wind speed, humidity, temperature, and surface area, that can either increase or decrease the rate of transpiration. So for this experiment, you're going to need these different materials. The key thing here is that the silicone tubing is a snug fit for your graduate pipette and the leafy stem that you choose. You submerge these items underneath water and try and keep the leaves of that stem out of the water. The diameter of the tubing is really important. Here we've used 4 millimeters, which I find works really well with horse chestnut tree leaf stems, but that has to be a very snug fit with your pipette and your leaf. Here you're going to use a pipette or a syringe to remove any air from both the graduated pipette and the tubing. And when you're satisfied that there's no air in either of those, you can fit the leafy stem into one end and the graduated pipette into the other, with the top of the pipette into the tubing itself, and you can seal that with some Vaseline. You then mount that into a retort stand as shown here, and you can position that wherever you like based on your independent variable. And over that period of time, you're going to track the movement of the water level through the graduated pipette over a period of time, and your results might look something like this. So over the course of 30 minutes, that 1 ml pipette was drained. This slide shows you how you might adjust those different independent variables for this particular experiment.
Okay, right, so I've been asked about environmental science. I don't think I would be the best person to do that. I know that they're based on, I can never say the word, but the equivalent of synapse for environmental science, that is Latin for squirrel, I can't say it. They've got something big going on. So they've got some kind of big webinar, and I think that sounds like it's going to be really good. So if you're not on that, I can send, maybe email me about it and I can pass on what I can find out about it. But there's definitely some key people in Edinburgh that are organising something. Okay, so these are the pictures, also really lovely to be able to get. Yes, that's the name of it there. Yes, good. And like was in that video there, the kind of data, and it goes really quick, it's really nice. And as long as you get the seal right, which we had a group here who did it recently, and I think only one of them leaked a tiny bit once, and then it was quickly resolved. So it's just getting the seal. And I found, although some people are lacking in the tree department, that actually horse chestnut leaves were the best. It was something about almost like how uniform the diameter of the stem was, whereas with laurel and other things, they could be almost too woody to securely fit into the tubing. So that was my preference, and I found because the span of the leaves, it worked really quickly. So thinking back, oh, I thought there was another slide in there. Oh yeah, there is. So now moving on to 3B. So now this is focusing on, was there enough raw data? That's all we're looking at. Were there sufficient repeats? And that depends. It's not an absolute here. It depends on the practical you're doing. So something like enzymes, then cool. 3 repeats is usually fine. If you're doing something out in the field with quadrats, my experience is they're looking round about 10, sometimes 5. I know with things like Daphnia, they say 5. Quadrats or field stuff, quite often 10. If it's things with humans, so there's one in the Higher Human website that recommends the serial position effect. I've heard big numbers when they've talked about how many repeats they're looking for. So that might be worth an Understand Standards clarifying. For that, if anyone else is sure, please add in. So here, all we're looking for were the number of repeats sensible and appropriate. So if this was the data, say you had surface area of leaf, you had 3 different, your range was between 20 and 40, and you did 3 repeats. The key thing I'd be looking at, which I think is a big ask for National 5, but is there a lot of variety there? They're really reproducible numbers. 3's good. If they're all over the place, then you would go, oh, it's not very reliable. Maybe I should do a bit more. So I would probably be driven more by that. But I do think that's a big ask for National 5s to relate to. If you were wondering, like I was, how do you calculate the surface area of a leaf? This link here brings you to a pretty good website on how to work that out. Really straightforward. I started to have horrible images of like working out the area under a curve like the physicists do, but that link is nice. So sufficient number of repeats there, good. Is the range appropriate to them? Good. So here they would get their mark for that. For C, then we're looking at, is the data presented in a correctly produced table? Is it tabulated with the correct headings and units of measurement? And every column must have a clear heading, and that's probably an important part to pick up on. And units can be in the heading or in every data entry. So if this was the example, you might look at that and thinking about that sort of guide there of the mark instructions, think, is this good or not? The answer is no, because there's no units on that top column. So that wouldn't get a mark for this particular part of the marking scheme. Again, this one is all good. That would be an example of a good table. This one here is bad table. And this is a good example to pick up on, because SQA really emphasised this one when I went to a markers meeting, because apparently, well, it is done quite a lot. I didn't find it was done as often as, I didn't think it was a very common mistake, but making the average column completely separate and not under volume of water transpired is something to look out for that they might do. So that would be not good. And the other thing which they were saying there is, if, I don't think you can probably see my arrow there, but where it says surface area of leaf centimetre squared, if they didn't put it there and put it beside the 20, 30 and 40, they would also be good. Okay, so that's all that I had for that one. And then, have they calculated the means correctly? So here, if you actually, and marker has to genuinely go through all of the repeats and calculate the average and check them. So these are all good. When you've got time on your hands, you would check those and go, yep, those are all good. So keep an eye on the average that is there, so 0.29. If they said 0.28, that is a rounding error, and that would not get any of their marks then. They would lose that mark based on that. And the other thing that is really common is that you work out your average by dividing by 3, and you get 0.286, blah, 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 blah. If they add more numbers after the decimal point than the raw data, then they have implied a degree of accuracy that the raw data can't provide, and so they would lose the mark. So if they've gone 2 decimal places in their raw data, their average needs to be 2 decimal places as well, is my understanding. Okay, right, we'll go to 3. I feel that I am quite slow. Effect of something on pectinase activity is the next one. Okay, so here you could do that range of different independent variables. I've just done in this example, yes or no. Does it have pectinase? Does it not? And what difference does it have? And it's a really lovely, simple, easy to do experiment. Again, it would probably work at higher human, but you would have to do a bit of adaptation for the range of different independent variables there. Good question. If the raw data is to 2 decimal places, can they bring it to 1? I don't know. I'm not sure. My gut feeling would be maybe, which is useless. I don't, oh no, thank you. Thank you. Someone else can, with more experience of that, kind of, okay, I would have thought that would be okay, but I'm glad someone definitively knows that. Okay, so the background for this is in all these next slides, which I'm going to skim over. But when we're trying to extract juice from fruit, we can manually do it if you fancy squeezing an apple for the whole night, or you can use an enzyme. So you've got different ones that commercial industries might use, such as pectinase, amylase, and cellulase, and they're all targeting different components of the cell wall. So it might be that, I think it's in this one, so the pectinase is breaking down the pectin. The cellulase, unsurprisingly, is the cellulose and the hemicellulose, and the amylase is just generally working on some of the more complex materials in there. And as we break those things down, it allows that juice to become, you extract more and it becomes clearer. So it then increases the profitability of that industry. And another thing that you could do here, which we're not going to show you, is immobilizing the enzyme. So you could do that here exactly the same as we looked at in the first one. Okay, so I will play this one. Really lovely and straightforward to do.
Okay, so in this experiment, we're going to look at investigating the effect of pectinase, an enzyme, on juice extraction from apples. And this is an example of an assignment topic that SQA have recommended at National 5 level, with various types of independent variables that you might wish to investigate. Arguably, this could also be pushed up to higher human by looking at enzyme concentration, for example. This is an overview of what we'll do. So we'll get the apple, we'll blend it to fine consistency. We're going to add water or enzyme to that mixture, incubate at 60 degrees, and then look at the volume of juice in each of the different reactions. We need a variety of different materials here. You can choose different types of apple. In this particular example, we've looked at Granny Smith, and we're looking at the role of pectinase as the enzyme. So you're going to collect two 20-gram chunks of apple. You're going to blend it to a finely chopped consistency and put it into two different beakers. To the one on the left, you're going to add water. To the one on the right, pectinase, and in both cases, it's 5 mils. And then you're going to incubate those at 60 degrees Celsius for 10 minutes. After that, you're going to take two measuring cylinders, about 25 cubic centimetres, and add a filter funnel to the top with some filter paper. And you're going to decant those mixtures into the separate measuring cylinders to look at how much juice has been extracted in the presence of water or the presence of pectinase. And in those filter funnels, you can see that there's quite a lot of apple left over. So we also want to look at what mass of apple has been wasted in this process. So this is with water, and the second in the presence of pectinase. And you can see that pectinase has made a big difference, both to the waste apple and to the volume of juice extracted. This might be example results that you might see. This is after a 20-minute incubation period. And this is it in graph form as well. So on the left, volume of juice, and on the right, mass of waste apple.
Okay. It's always good having to sit here and listen to your voice in a video. It's my favourite thing. Right. So here is the data for if you just were looking at volume of juice extracted in the absence of enzyme and then in the presence of pectinase. And you could maybe extend it by looking at different types of enzyme. If you've got cellulase and amylase there, concentration of the pectinase, there you have to do it like a percentage, because I'll give you the kit list at the end. We always go to NCBE for this, and it doesn't tell you the concentration. It's just like, this is 100% pectinase, but that's absolutely fine. Okay, so thinking about how you would maybe target some of the aspects of that for 4, which is where we move on to yes. So everything would need to be repeated with averages. Yeah, it would. So let's look at 4A. So how would you graph this? So if this was the data set, so say we looked at pectinase concentration and you did 3 trials, you had some nice low levels of variability there. So you did it 3 times and you've calculated your average there. So here, where we're talking about those decimal places, so this is 3.0, 9.0. If they don't add the 0.0, my understanding, and I could be wrong, so please do feel free to correct me. If they don't add the 0.0, that's okay. But if I'm wrong, please do say so. But that's what that would be, 3.0, 9.0. So in that particular example, we have got continuous and continuous data. And so we would expect that a line graph would be produced, and you'll know the problems across the sciences in terms of graphs and assignments. But we would be drawing it like this, and I'll come to the Excel issue in a second, but it should be a line graph, and that's all they're getting marked for in 4A. Have they done a line graph? Okay. Right, okay, thank you. What they will say is that hand-drawn graphs are a safer bet. So they have to really know how to use Excel to put the minor and major grid lines onto an Excel graph if they're going to go down that route, because otherwise they can't determine how accurately those points are plotted. So it is safer just to go down a hand-drawn graph way. They also don't like that using data from, right, okay. B, the axes on the graphs have suitable scales. So in this case, Excel has done the hard work for me. If your pupil's graph looks like that with those scales, that's good. They start at 0. Last time I was marking, they accepted a common 0. They didn't have an issue with that year. That may have changed because ordinarily in biology we wouldn't do that. But they really loosened up in that last year about the axis in terms of the scale. So the axis of the graph have suitable scales there, so that's good. They're a linear scale in both directions. What wouldn't be okay is this one at the side, which is really common if they do pH. So if they've got pH 2, 5, and 12, quite often they write 2, 5, and 12. And obviously then it's not linear and they don't get their mark. Here, last time SQA said this was okay because there's no 0, so you would be worried, but they said that was fine. And actually they went further and said, as long as there were 3 points that implied that it was a linear scale, they were happy. But I would obviously be very cautious about that. But they were much more lenient about scales last time when I'd marked for them. So I don't know if anyone else has got any comments on that. In terms of the axes have suitable labels and units, that's what 4C is looking for. Spell mistakes are okay. Abbreviations can't be penalised if it's obvious what they mean by the context. So here they would get the mark. Notice that I've missed off average from the y-axis, but that's okay because it's obvious from the data they've plotted that they've taken their average data. The units are included, that's good. If they'd said vol or conc, that would also be okay, but generally, obviously, it's good practice just to copy and paste from their table. That's the same guidance for the graph as the exam. I think there's an explained format in the candidate guide. Okay. Okay. So yeah, okay. So there's some chat that's sort of seeming like I've not said a whole bunch of rubbish there, which is good. For 4D, then we're looking for accurately plotted points. And again, here I would emphasize that hand-drawn graphs are better. I just didn't want, I probably was being lazy and didn't want to hand draw it. But that would be better. So if using Excel, we need the grid lines. We need to make sure that we can see all of them and they're not the default really, really light ones. Don't penalise. They don't penalise for plotting an incorrectly calculated average. So if they've calculated it wrong and then plotted that wrong value, that's okay. And when they hand draw it, they allow a half box tolerance for hand-drawn points and tops of bars, although there was some discussion on Synapse a while ago about whether or not that was accurate across the levels and what different people felt about that. But that was my experience. Okay, so we're going to move on to the last one, which is looking at how do we look at the effect of something on photosynthesis. So let me just check I've not missed anything. No. Are they penalised twice for having an incorrect heading in the table and label on the axis if they're both the same? My understanding for that would be no, because the whole principle is that they would never penalise twice. So if they've done it wrong once, that's fine, Chelsea, no worries. If they've done it wrong once, they would get penalised once. And if they've copied that heading over to the graph, my feeling is that they wouldn't get penalised twice. Okay, so let's think about photosynthesis. So we know photosynthesis is how we're going to convert light energy into these chemical bonds, and that light intensity will affect that, and also the wavelength of light will affect that. So we've got all these pigments that are going to absorb the light, but they will absorb optimally at different wavelengths. So for this particular protocol, we could look at the effect of light intensity or the wavelength of light. And because of that, we can either look at National 5, and we are going to use bicarbonate indicator and algal balls, and we could look at this, or at higher, they also recommend looking at wavelength of light. Okay, we are using Senidesmus. I'm not even going to try and say the second word because I'm really bad at doing things like that. But this is a really good organism to immobilize. And you basically grow it up, so it's really, really concentrated. You can spin it down, and then you take a little teeny weeny bit of that and you immobilize it with sodium alginate in the same proportions that we used in the yeast protocol at the beginning. So exactly the same thing. So across these different visible wavelengths of light, depending on what we allow plant like algae to have access to, it will photosynthesize different rates. So we use a range of different filters if we were looking at wavelength, which I've given you the link for where to buy those. But we do that because we can get the full spectral properties of those filters. And so, for example, with the blue one, it tells us that the maximum wavelength of light that the algae would have access to is at 455 nanometers. With the green one, we use 530. And the red is much more tricky because it's much broader. So I kind of just tell a bit of a fib and say it's 690, but it's tricky to pick from that spectral spectrum there. In this one, we're going to soak in or incubate, that's the better word, the algal beads in bicarbonate indicator. And what that does is, as carbon dioxide is used up, we will move from this acidic concentration, this more yellowy colour, and we're going to move to the purple because we've now got less carbon dioxide and it's less acidic. So the more purple, the more photosynthesis is taking place. And in this picture, and in a lot of the pictures I've had, it's seaweed that we're working with, and you could do that as well, or you can immobilize the algae. So whatever you have, or you can use the Duria. There's lots of different things that you can use. As long as it's an aquatic plant, it's fine. And if we're using algal beads, in this top row, you would have something just with indicator, and the rest of them have indicator and the beads, and you would either have access to full light or different filters that change either the light intensity or the wavelength of light going through to the plant. You're then, after a particular incubation period, you're going to then decant that indicator into these cuvettes and you're going to measure the absorbance. So this is the last video here.
In this video, we're going to be looking at an investigation for looking at the effect of light intensity or the wavelength of light in photosynthesis in algae. And the underlying biology from this will look at the background to photosynthesis, different factors that might affect the rate of photosynthesis, and might even start to look at the different pigments that are involved in absorbing the light energy, such as chlorophyll A and B and the carotenoids. And here are our possible aims that we might be looking at. Light intensity may be more applicable at NAT5 and wavelength of light at higher. Both of these are suggested by SQA to use, and that's NAT5, and again at higher, both using immobilized algae, but potentially looking at light intensity at NAT5 and wavelength of light at higher. We're going to use Senidesmus in this, and we're going to start by immobilizing the algae. These are the materials that we need. So immobilize our algae, immerse it in bicarbonate indicator, and then we're going to read the absorbance using a colour emitter. We're going to change the wavelength or intensity of light using these filters. And from Lee filters, you can have a look at the specific spectral properties of these different filters that you can apply. Now, bicarbonate indicator will change from yellow to purple with decreasing levels of carbon dioxide. So if photosynthesis is proceeding really quickly, we should progress more purple in colour over a shorter period of time. This is an overview of what we're going to do. So you're going to have 5 bijous, you're going to add about 6 mils of indicator to each, and 20 algae beads to 4 of those. We're going to add the different filters, incubate for 30 minutes, and then measure the absorbance of that indicator after that period of time. To immobilize, you're going to add the concentrated semidesmos to sodium alginate. You're then going to pass through a 10 ml syringe with no force from the plunger, just to allow that to drop into a beaker of calcium chloride, periodically giving it a swirl to allow those beads to form. And when they all sink, that's then ready to be rinsed in distilled water and used in the experiment. You're going to collect your 5 bijous, and here it shows seaweed, but you're going to add 20 mobilized algae beads that have been rinsed into 4 of those. You're then going to place your different filters, whether they're neutral density for light intensity or coloured filters for looking at wavelength of light, over the last 3. We're going to then place them in front of a light bank and allow the light to penetrate through those different filters for 30 minutes. And at the end of that, remove the filters, remove the beads, and decant those the indicator into separate cuvettes and measure the absorbance of light through those. If we were looking at neutral density filters and the effect of light intensity, this might be similar results to what you find. And in this next slide, we can see what we might expect if that was looking at the effect of wavelength of light on the rate of photosynthesis.
Okay, let me try and pick up on some of these things that people have asked here. I don't watch any of these things in my spare time. I don't know why it's recommending these unusual videos there. Think you can provide it, right? Okay, so how do you standardize the amount of seaweed? Yeah, so when we've done that, we've done things like mass of seaweed was a really weird one because it depends on how hydrated it is. So one time I went out and thought, right, 1 gram of seaweed is good. Next time 1 gram of seaweed was like a huge bundle of it. So we go for length of seaweed, and then you have to avoid all the air bladders because they're full of gases. So yeah, length I think's okay. And then you've got thick bits and thin bits. I would just try and keep them as in the same general surface area, I guess, would be a good way of going. Okay, so these might be the type of graphs that you might see if it was for light and for wavelength of light there. Okay. Okay, so candidate 2 from National 5, this is what they did. So again, you've got a really nice bit of data. Oh no, it's from higher, sorry, to actually go back and have a look at. So yeah, that's candidate 2 on the Higher Understanding Standards website. And so I'm not going to go into it. I don't know enough about the Higher to say any more about other than what I put on the screen here. So with the aim, if their aim was this, so to investigate if increasing the percentage of light intensity would affect the rate of photosynthesis, how do they, so with their second source, how do they do a valid comparison of that? And this is a really difficult bit. So their data look like this, where they have got percentage of light intensity and then their average. And so as the carbon indicator increases in pH, it means that there's a higher rate of photosynthesis. So they could roughly say as light intensity increased, their rate of photosynthesis increased. Their second source looked like this, which is really tricky to be able to see, but they've basically got pondweed and they've moved it closer and closer and closer to the plant along this way, and then count the number of bubbles that they get. So it increases and then levels off. So how did they compare it? So our source and my chosen source are not directly compatible because they looked at different things. However, internet source looks at number of bubbles given off. However, they are not directly compatible, but they do show that as you increase light intensity, the rate of photosynthesis also increases. And that's a really common thing for them to say. They don't get the mark because they don't talk about the differences in the trend. So it's not as straightforward as they have said. So here, what they've looked at, the independent variable is different and they needed to comment on that. And they haven't talked about the full trend. And that's why they don't get the mark there. And it's the same, in a way, it's the same issue carried through to the conclusion. So they've got to write the conclusion based on everything they present, so from their data and source 2. And what they say again is that the higher the light intensity, the higher the rate of photosynthesis. So source 2 doesn't show that. And so the conclusion is not valid because it's only supported by their experimental data. So that's actually all I was going to go through. I know that I'm actually really approaching the end of time. The Q&A, I have tried and done a really terrible job of keeping an eye on your chats. So I'm really sorry, but I think you've sort of helped each other out a lot. If there are questions, then you can go back into that and I'll do my best to go through them and help wherever I can. And then otherwise, I'll pass on what I think are the questions for SQA to maybe answer. And they, like I say, they say they're going to address those in the Understanding Standards events. Scan this now, because that will give you your files to download. I am going to email out, but emailing out this number of people is really difficult because it bounces back, I think. There's some issues there. So if you don't receive things by Monday, I'm away tomorrow at Napier, but if you don't get them by the end of the day on Monday, email me and say, Annie, you've not done what you said. And I will send you, it's just a link to the folder that's got everything in. Okay, so if you don't get them, please do get back in touch with me and I'll fire in the recording as well. And otherwise, I have to put the evaluation up. I know that lots of people don't like doing evaluations, but in case you wanted to give any useful feedback, then that's another QR code. Let me just make sure I gave you everyone enough time. Yeah, I think microscale would be okay. I have thought about that. But yeah, I'm just, I'm reluctant because SQA put their list out, but my feeling is they can do that quickly. You're still getting data that allows you to fulfill, as long as you can fulfill all the points or the aspects of the mark instruction, it should be fine. Right, that's me. I can probably stop sharing now if I can remember how to do that. But yes, please do feel free to head off and get your dinner and your children and all the other things that you have to do now. And I will email those things out. Can you put up the QR code, put a previous one? If that is for, let me just get the link for the folder itself, and that might be easier for everyone. So the folder, oh my goodness, this is so difficult with so many things happening. Right, let me see. I cannot access the chat now because of all this stuff. Right, wait a minute. Right, okay. So this is the link to get directly into the folder, I think. Is it coming? Yes. So that should get you into the folder unless you've already left. If a student plots one pointing, yes. So that is the worst. I remember marking ones where they had looked at about 30 different things. So they'd really gone to town and they'd done so much work and one of them was plotted wrong. I think it's my most hated thing because it's such a shame and it doesn't reflect their ability at all. But that's what it was before. So I always said, try and keep the number of points you're doing small. I didn't catch the QR. Right, so Heather, I've put the link in. If you can see that, I can share my screen again though, just in case. Let me fire that over here. So that's the one that you need for the files. Cats have watched. Oh, good. I'm glad your cats have watched, Kirsten. I always like to be a friend to all pets too. Let's see who all's still here. So I don't think I've missed anything. Can you put the link up instead? Yeah, okay. Okay, I think I've got everything there. Right, okay. If you're still here, have a lovely, lovely evening, and I'm going to leave imminently. But if you've got any questions, please come back to me at any point. And goodbye to all the people that I recognise. Such a shame I can't see you. Oh. Yes, you have a good evening too.